In this lecture, we're going to be starting a unit or a section on the cell. Most of us are familiar with a typical drawing of an animal cell, where we have on the outside a cell membrane that surrounds an internal space. Often it's drawn as a round circle, and then inside of the cell we know that there's a nucleus in there. That's something that can often be seen with a microscope when you're looking at the cells, and sometimes inside the nucleus you'll see a little dot inside there called the nucleolus. So those are the main things we can see typically with a microscope, is the membrane, the nucleus, and sometimes the nucleolus. Usually the internal structures of the cell would not be visible, the things we call organelles. So we're going to go ahead and start talking about all the different features of the cells, different variations of cells, and so forth in this next section. While I drew this as kind of a cuboidal cell, like a square kind of cell, sometimes they are rounded, like in most blood cells, they're more rounded in shape. Sometimes cells are flattened, flat as a pancake. Sometimes they're drawn out in spindle shape, very long and skinny, like a smooth muscle cell. And so anyway, there's a lot of variation in shapes, and we'll talk about kinds of villi and microvilli, little extensions or unusual features of the cell surface involved there. So that's our next thing to do. Now, to outline some of this, the cell overall structure here, we've already drawn the diagram. The cell membrane surrounds the outside. Cell membrane is also sometimes known as the plasmalemma. Sometimes it's called the plasma membrane. So all those are the same thing. So that's that outer covering. We'll look at that in detail later. Cell nucleus, the nuclear membrane, also known as the nuclear envelope. Lost my E there. Nuclear membrane or nuclear envelope surrounds the contents of the nucleus. And inside the nucleus then you have chromatin, which is of this stringy mass made up of the chromosomes. So the chromatin and the chromosomes are essentially synonymous. And then finally the nucleolus, a structure inside the nucleus that has a special function for the production of ribosomes in there. So we have the membrane, the nucleus, and then finally the cytoplasm. So next we're going to talk about things that are in the cytoplasm in our next outline. Cytoplasm was just another name for cell plasm, this material that's kind of gel-like or not exactly fluid, that's between the nucleus and the cell membrane. Everything in that space we consider cytoplasm. It's made up of a component we call cytosol, the fluid portion with different solutes, ions, and other things dissolved in there, or suspended molecules within that fluid, the cytosol. And then in addition to that, just general mix of chemical mix of fluid inside the cell between the nucleus and the outer membrane. We have the organelles, functional components of the cytoplasm, different kinds of structures with specific roles in the cell. Depending on the textbook that you use, that you pick up, you'll see sometimes that among the organelles we do include things of the nucleus, so the nuclear membrane, the chromosomes, and nucleolus. Those features could also be considered organelles by most textbooks, and that's kind of the way I think of it. I always include those things as organelles when I'm talking about the cell. So next. Now the cell membrane or plasmalemma is made up of what we call a phospholipid bilayers. The phosphate heads are the brown circles here, and then the fatty acid tails, two green extensions coming off of each of the phosphate heads. So this single one here would be a phospholipid. A single phospholipid with a phosphate head. Phosphate head is what we consider a polar molecule. That means that there are some regions of this structure that have fewer electrons on them that are more positive, and other areas more negative charge. So it's not a electronically neutral structure with respect to water specifically. The fatty acid tails are nonpolar. They do not have a charge in here. These are fatty acid tails. We just call them tails. They're not anything special. We could just refer to them as fatty acids attached to a phosphatase structure. But anyway, it's easier to think of them as heads and tails. So these are nonpolar fatty acid tails. They are nonpolar. Nonpolar fatty acid tails tend to make associations with each other. Same way that oil doesn't mix with water, these nonpolar elements in here do not make associations with polarized molecules or molecules with polarized regions on them. So we have the nonpolar region in here forming the core or middle portion of the membrane, and then the phosphate heads on the outside. So we have polarized regions toward the water. It's either to the inside or the outside of the cell, and nonpolar elements, the fatty acid component, on the inside here. In addition to the fatty acid tails in there, you'll also see cholesterol molecules in between here. So you'll find that, can't really just draw a little cholesterol molecule in there, another cholesterol molecule over here. So they kind of get embedded within the lipid portion of the phospholipid bilayer. Now there's an outer surface of the membrane toward the outside of the cell. We consider that the external face, and that layer of phospholipids right there that makes up that outer component right there is called the E face. E face for external, the external face. And then the other one is against the plasma of the cell, the internal cytoplasm, the plasma of the cell, and that is referred to as the P face. P for the plasma components inside, E for external outside. So there's an E face and a P face on this phospholipid bilayer. In addition to these components, there are proteins and glycoproteins and glycolipids, which we'll get to next. Now we're going to add proteins to our phospholipid bilayer. Some of our proteins will take up a position to the outside. They'll lie on the external face out here, so let's put a protein structure in over here. They lie on the outside. There are some that are embedded within the E face phospholipid layer. Different shapes of proteins, different kinds of proteins can be embedded here in that E face portion. There are what we call transmembrane proteins that can go all the way from the inside to the outside of the membrane, so transmembrane proteins. The next position would be embedded within the P face, the internal face, here. Just making kind of random shapes. So those are 4 different positions, and finally we could have them lie on the inside. Against the inner surface of the membrane. So those are your 5 different positions. So you have your external surface, you have embedded in the E face, you have transmembrane proteins, you have embedded proteins on the P face, and then you have internal surface. Proteins, those are your 5 different positions those membrane proteins will occupy. We'll talk about functional categories of proteins here in just a little bit. But anyway, the next thing we need to add to this, and I don't have in this drawing any of my cholesterol, but remember there's cholesterol also within, between some of these fatty acid tails will be some cholesterol in there. So we have cholesterol, we have the phospholipids, we have proteins. The next thing to add is what we call the glycocalyx. Glycocalyx. There are some proteins and some of the lipids of the membrane that will form a connection to some carbohydrate chains, and the carbohydrate chains occupy space to the outside of the cell. So here I'm not going to go to a great deal of trouble to make things seem very accurate in terms of their shape. A lot of 5-sided or 6-sided ring structures probably in the carbohydrate chains, and some of them are attached to proteins and some of them are attached to the lipid components of the membrane. So like I said, some of them are considered glycolipids. Carbohydrate chain attached to a lipid component of the membrane, and then there are glycoproteins. That are associated with proteins. Glycogen or a carbohydrate unit attached to a protein unit. Now they're found chiefly on the external surface of the cell where they form what we call a cell coat, which is the same thing as glycocalyx. Cell coat or glycocalyx. That arrangement of carbohydrates attached to lipids and proteins along the outside surface of the cell. So those are the things, the primary components of a cell membrane. Now we're going to identify 6 functional categories of membrane proteins. So functional categories of membrane proteins. Number 1, channel proteins. Channel proteins make an opening that passes through the membrane. These are always transmembrane type proteins. They go all the way through. But some of them conduct water. They allow water to pass across the membrane. And some of these are what we call aquaporins. Aquaporins are water channels through the membrane. Some of the channels will be ion channels, allowing ions to move from one side of the membrane to the other. These channels are often very ion specific. They only allow one type of ion to pass from one side to the other. So that's channel proteins. Number 2, carrier proteins, transporters. Another name for them is transporter proteins or carrier proteins. They grab a substrate molecule on one side of the membrane, and then they change shape or do something with the molecule to move it and release it on the other side. So they grab a substrate molecule on one side and then release it to the other side of the membrane. It can go into the cell or it can be moved to the outside of the cell, depending on the carrier. There are a lot of specific carrier molecules for specific kinds of molecules that need to be moved across the membrane. Next, receptor proteins. Receptor proteins will bind to a substrate molecule, some kind of molecule on the outside of the cell, for example, and the substrates are not going to be moved across the membrane here. They're just going to bind to it. Substrates which bind to the receptors are called ligands, L-I-G-A-N-D-S, ligands. So the very definition of a ligand is something that binds to a receptor protein in the cell. Next, moving on to number 4. Enzyme proteins. Enzymes are molecules that facilitate metabolic reactions. They help move things along to get molecules to change from one form to another, or to bind with another, or to be broken apart. So I have here A can become B, just a transformation of a molecule. A plus B can become C, combining molecules, or a substrate can be divided into two parts. A becomes two parts, B and C. So any of these things, these are what we consider metabolic reaction, changes to a substrate molecule. Number 5, linking proteins. These proteins form attachments to neighboring cells or matrix. Now, matrix is a term for things that are not cellular, found in the tissues, that make up that material between cells. So there's bone matrix, cartilage matrix, some places it's just collagen matrix or other kinds of fibers out there. But anyway, so you have different kinds of linking proteins, number 5. Number 6, cell identity proteins. They allow cells to identify neighboring cell types. So your tissues kind of work together so that epithelial cells will bind to other epithelial cells to form a layer and so on. Immune cells can recognize cells as self, so you know what's your own body and not a foreign material that you need to wage an immune response to. So some of the cells have these. When you talk about your blood types, your A, B, O blood types, those A and B structures, they're actual proteins on the surface of your blood cells that can be identified as foreign if you get the wrong kind of blood. If I were type O and I would receive type A blood, I might have a response to that, an immune response against that A-type molecule that was coming in. It's not part of my body. So those are our 6 different functional categories of proteins. Next, I'm going to talk generally about cilia and microvilli and stereocilia. We're just going to look at a couple of examples of drawings here, and then we're going to give an outline of the different categories and types of cellular extensions. These are things that project off of the cell, usually an apical surface of an epithelial cell that extend outward. First of all, cilia. This is a single cilium right here. So these are cilia. The cilia are made up of, within the membrane that surrounds it, is made up of a structure known as an axoneme. It's an arrangement of microtubule structures that are arranged in pairs that extend straight up and down through the cilium. Here I have the cilium cut in cross section, and there are 9 pairs found in a circle around the outside of 2 other microtubules on the inside. So this is called the 9+2 arrangement of microtubules within the axoneme here. There are more proteins involved in this than just this structure, but just to identify this as a motile cilium, a kind of cilium that undergoes regular movements, we want to know this arrangement, that 9+2 arrangement, 9 pairs of microtubules around a central, 2 independent microtubules on the inside. So that's the 9+2 arrangement. So this is the axoneme. This structure down here at the bottom, instead of being made of pairs down here, there's sets of 3 microtubules arranged in a circular pattern. And this is always hard to do, to get this to come out right. But anyway, there's 9 pairs of those. I only have 6 of them in my picture here. But anyway, 9 sets of 3 in this portion known as the basal body. At the base of a cilium. So what I've drawn here is a motile cilium. Now there are motile and non-motile cilium. So motile is the 9 plus 2. 9 plus 0 would be non-motile types of cilia. The non-motile varieties are there, kind of like cell antennas. They have a sensory function. So whenever you talk about cilia, you're talking about this kind of arrangement. Cilia are primarily within the respiratory epithelium, on the respiratory surfaces through your lungs and bronchi and bronchioles is where you'll find these cilia. They have this continuous movement that sweeps any kind of debris or particulates that you inhale, moves them up through the mucus, carries all the mucus with it, all the way up to the throat where you swallow it. Also found in your nasal passages, another place where you find that. Also found in the oviduct, where you have to generate a current of fluid through the oviduct to essentially catch the egg cell and make sure that it's swept into the right direction to end up in the uterus, to carry the egg cell to the uterus. So that's where you find your motile cilia. We'll talk about non-motile cilia here again in a minute. The next example here is a microvillus. The microvilli and the stereocilia both have the same kind of internal structure. Instead of having microtubules on the inside, we have microfilaments, also known as actin. Microfilaments, or actin fibers, protein fibers arranged in straight columns within the core of the microvillus in the same way that, well, not exactly the same way, but they're arranged up and down linearly, just like what we saw with the microtubules within a cilium. Completely different kind of protein structure, however, and they're not motile. These are usually absorptive. They're found abundantly in the intestine for absorption and in your kidney tubules, some places like that. So those are microvilli. So next we're going to a stereocilium, we'll talk about that as well. Stereocilium is actually a type of elongated form of a microvillus, much, much longer than this. So we're going to outline the different kinds of cell extensions, but don't forget this. This is the motile cilium example here, attached to a basal body. There are non-motile forms that do not have the pair of microtubules in the center, and then microvilli and stereocilia have actin in their internal structure. So for our first two forms of cell membrane modifications, we talked about the cilia and the microvilli. Just to put this on the board, the cilia contain an axoneme and the basal body. We saw that basic shape. The axoneme made out of the microtubule arrangements, that 9 plus 2 arrangement. The motile cilia are in the respiratory system and the oviduct lining, the lining of the oviduct, where you see that 9 plus 2 arrangement and moving cilia that actually conduct motion, produce motion in fluids overlying those epithelial layers. Then the non-motile cilia, a form of sensory antenna in many cell types throughout the body. They have the 9 plus 0 arrangement. They're not motile. They're simply an extension of the cell, and they give the cell information about events to the outside, usually motion or movement, displacement on the outside. But anyway, so that conveys information to the cell through non-motile cilia. So those are cilia that have the microtubule arrangements on the inside, that axoneme. Microvilli, they have microfilaments or actin filaments on the inside. They're very similar to cilia in that they're projections from the cell, hair-like or finger-like extensions from the cell, but they have these microfilaments inside instead of the microtubules. They function to increase absorptive surface area in the intestine and in the kidney. Now, next we'll look at a couple of other types. Next, stereocilia. We've mentioned that before. They're essentially very much like a longer version of a microvillus. They're not like a cilium, so that cilia portion of that is a little unfortunate, more of a stereovillus than a stereocilium. So stereocilia, a longer version of microvillus. They do have different proteins associated with the actin fibers on the inside of a stereocilium, and different types of proteins in different forms of stereocilia. We recognize two major types of stereocilia. There's a type found in the male reproductive system primarily, but not exclusively, in the epididymis of the male reproductive system, that are considered to be absorptive or thought to be absorptive. They remove fluid, water from that material around the sperm. And then the other type are sensory in the hair cells of the inner ear. They're associated with the cochlea, with the semicircular canals, and with the vestibular portion that gives us our sense of static equilibrium or position with respect to gravity in there. So we have sensory in the inner ear and absorptive in the male reproductive system. Our next type of cellular extension would be what we just consider a fold, a flap-like extension from the surface of the cell. We also call these plications. Plications, they are found either on free surfaces or interdigitating with neighboring cells, so that the folds kind of interlace with one another. It's like having two curtains next to one another, that their folds follow each other, interdigitating with neighboring cells. These folds can form outward folds that go out, or the folds can go toward the middle of the cell. It can be inward reaching or outward reaching folds or plications on the cell. And then our final category of extensions from the membrane are internal extensions. We call transverse tubules or T-tubules. These are found in muscle fibers. Tubular extensions of membrane that extend to the inside of muscle fiber, and when we get to looking at skeletal muscle, we'll show what those transverse tubules look like. They're just like straight tiny tubes of membrane that run through the muscle. When we look at the cell organelles, there are several reasons why we might want to divide it into non-membranous and membranous organelles. Here we have a column of membranous organelles. Over here we have the column of non-membranous organelles. Among the membranous organelles, we have the nuclear envelope, which we've already mentioned we could call the nuclear membrane, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi, sometimes called a Golgi apparatus, or a Golgi complex, or Golgi body. Anytime you see the term Golgi, it all refers to that same structure. Lysosomes, mitochondria, and peroxisomes. So those are membranous structures of the cell. Non-membranous of the organelles of the cell we're going to include chromosomes here and the nucleolus, ribosomes, proteasomes, which many people are not familiar with at all, microfilaments, which we know to be actin. It's the only kind of protein involved in the formation of microfilaments. Intermediate filaments and microtubules. These last three here are all involved in forming something we know as the cytoskeleton. And microtubules are involved both in the formation of the centrioles of the centrosome and in the formation of the spindle fibers at cell division. So these are your cellular organelles. Cellular organelles.